What are the costs and benefits for a pathogen of becoming resistant to antimicrobials? How can this lead to the emergence of antimicrobial resistant pathogens? How may modelling and genomics help inform the use of antibiotics in a way that reduces or prevents the spread of antimicrobial resistance? We explore these questions and more in this episode of SBIDER Presents.
This is SBIDER Presents, a series produced by the SBIDER Podcast Hub.
My name is Laura Guzman.
And mine is Ed Hill.
Today we welcome our SBIDER group member, Prof Xavier Didelot, a professor of Statistical Epidemiology and Genomics in the School of Life Sciences and the Department of Statistics at the University of Warwick. Xavier also leads the Health Protection Research Unit in Genomics and Enabling Data, which is funded by the National Institute of Health Research for 2020-2025. This project is a partnership between the University of Warwick and the UK Health Security Agency in collaboration with the Centre for Genomic Pathogen Surveillance and the University of Cambridge.
In this episode, we'll be discussing the research article estimating the fitness cost and benefit of antimicrobial resistance from pathogen genomic data. The article was published in the Journal of the Royal Society Interface in June 2023.
Welcome to SBIDER Presents, Xavier. We're happy to have you here.
Thank you very much. It's a pleasure to be here.
Great you could join us today.
Antimicrobial resistance is a major threat to health and is responsible for an increased risk of death and prolonged hospital stays. The paper considers fitness of pathogen variants. Could you explain to the listeners what fitness means in this context?
So, firstly, on the problem of resistance, I think it's a problem that many people nowadays are aware of. But just to give you an idea of how big a problem it is, it's been estimated recently that resistance of bacterial pathogen costs something in the area of 1 million deaths per year. And if nothing is done, it's estimated that by 2050, this could grow to 10 million deaths per year. So it's a really big growing issue, a major threat to public health, really. And so what we've been studying in this paper is the fitness of the resistant lineages. So what that means is that we try to compare how much of an advantage or disadvantage the resistant lineages have compared to equivalent bacteria that are not resistant. So we know that, of course, if an antibiotic is used a lot, then being resistant, you can see from the point of view of the bacteria, is going to be a good thing because the bacteria is going to be able to escape treatment and not die when the antibiotic is used. So there's a clear fitness advantage to being resistant from the point of view of the bacteria. But there's also a fitness cost in many cases, and we see that from the fact that if we stop using those antibiotics, after a little while, we see the resistance level go down little by little. And that's because the resistant forms of the bacteria actually have a little disadvantage compared to the susceptible ones, such that if the antibiotic is not useful and it's not useful to be resistant, then the resistance sort of goes away little by little.
That's brilliant. Thank you for that overview. So as a result of the COVID-19 pandemic, we've seen there's been an increase in public awareness of both mathematical modelling, data analysis, and also pathogen genomics, and its usage continues to increase. So for this particular research article, what were the knowledge gaps at the time that you were hoping to help address?
Yes. So the pathogen genomics is a sort of main type of data that we use in this study. And it's very, very useful for us here because looking at the genomes provides, in fact, a window into the past evolution of the pathogens in a way that we can't observe otherwise. So it's a little bit different for COVID, of course, because from the moment the COVID crisis started, there were a lot of scientists working on it and a lot of scientists doing surveillance and looking at the dynamics of the COVID epidemics with different variants appearing, etc. But it's not so much so for bacterial pathogens. Up until the 1990s or before that, surveillance of resistance in bacterial pathogen was not so well thought through, especially in some countries. So, but having genomes allows us to look at the past. Even if the genomes are collected only today, looking at those genomes and looking at what mutations have happened in those genomes in the past allows us to estimate what are the past dynamic of the different lineages of bacteria that we look at.
So you look at pathogen evolution for resistant and non-resistant
That's right. So in this study, we compare the genomes of bacterial forms that are resistant to a pathogen, to an antibiotic. We look at many genomes from bacteria that are resistant, many genomes from bacteria that are susceptible, and we compare their past evolution through by looking at what mutations we see nowadays. We can say something about their past evolution, and we compare their past evolution in a way that tells us if the resistant at one point was if maybe the resistant lineages were growing more than the susceptible ones, or vice versa. And we can correlate that with what we know about when the antibiotic was being used. And so what we see, of course, is that when the antibiotic was being used a lot, the resistant forms were growing more compared to susceptibles. And then vice versa, when some antibiotics were sort of stopped being used so much because they were becoming less and less efficient, then we see that the resistant lineages sort of start to disappear. So that allows us not only to establish this link between consumption of antibiotic and evolution of resistance, which is a link that, of course, we knew happened, but we can quantify exactly how strong is this link. We can quantify exactly what are the fitness, costs, and benefits of resistance in a way that now allows us to make predictions about how much of an antibiotic we can use without taking the risk of more resistance arising.
Were there novel methods or enhancements to existing methods required so you could attempt to do this estimation of fitness, cost, and benefit of antimicrobial resistance from this pathogen genomic data?
Yes, absolutely. So we had to build a whole new model for what we expect the dynamics of the resistant and the susceptible lineages to be. So that's an epidemiological component of the model. On top of that, we had to build a phylogenetic component, which represents how the genomes are going to, how the evolution of the genome is going to be affected by the epidemic dynamics. And all of this we implemented into a new piece of software that is able to infer the parameters of the model given what we observe as data, which in this case is the genome that we have of the susceptible and the resistant forms of the bacteria.
That's really cool to hear about the new frameworks that are being brought online to tackle these problems. And another key contributor or key component of the analysis would be the data used. And so what were the key data sources that were required to conduct this analysis? Were there particular attributes that it had that made it well suited to this research question?
So in terms of data, the first thing we did was to use a lot of simulated data, a lot of data where we know exactly that the advantage was X, and then we can test if our software is able to correctly reconstruct this advantage, say, of the resistance rate. So we did that a lot in order to prove that our algorithm works. And then, of course, we wanted to apply that to some real data sets. So we applied it to, in this case, a data set of more than 1,000 genomes of Neisseria gonorrhea, which is the bacteria that causes gonorrhea. It's a very nice data set, sampled over about 10 years from the United States, more than 1,000 genomes. And we focused especially on those lineages within that data set that were resistant or not to an antibiotic called ciprofloxacin that used to be used very commonly against gonorrhea, and is not so used anymore nowadays because there has been so much increase in the resistance rate against ciprofloxacin in gonorrhea.
Hearing about the verification of the model through simulation has made me wonder about if we went back a decade, 2 decades, would this have been feasible? Is there a need for a lot of computational power to conduct this type of analysis?
There is, yes. So that's a very good point. So there's a sort of all of this, of course, is implemented in software. So even if the models that we use could have been perhaps they're not that mathematically complicated, so perhaps they could have been sort of devised 10 or 20 years ago, no one would have even done it because the models would not have been applicable to data in the way that we do it. So for that, we need some very efficient computational algorithms. So in this case, we use an algorithm called an HMC, a Hamiltonian Monte Carlo method. So that didn't exist much before, about 10 years ago. And it's a very scalable algorithm that allows us to handle very large amounts of data, like what we have here. And we also have to run our software on some computer that are sort of the current power of computers, of course, is much more than it was 10 or 20 years ago. So for that's the other reason why it would not have been possible to do this type of analysis a decade ago.
And this type of analysis you can use with now different pathogens data sets.
Absolutely. So of course we looked at that example in gonorrhea, but really the model itself has nothing in it that is specific to gonorrhea. And we very much hope that it will be used by other researchers in the world to study the dynamics of resistance of different bacterial pathogens. There's a lot of different species of bacteria for which resistance is a growing issue, and so we hope that our method will be applicable to them and to different systems, and will help us understand better what are the resistance costs and benefits of resistance to a whole range of antibiotics for a whole range of bacteria. In that way, help us inform what is the best way to use the antibiotics that we have in a way that doesn't cause more resistance in the bacteria. So we know that more and more antibiotics are becoming at threat of not working. We know it takes a long time to develop new antibiotics. So of course there's a call for new antibiotics to be developed, but that takes a really long time from the moment of the initial research to the moment when the antibiotic can actually be used for clinical purposes. That takes a really long time. And there's, for that reason, over the past 10 years, there hasn't been that many new antibiotics being discovered and being brought to the clinic. So really, the other side of the coin, if we can't develop more antibiotics, is to try to make sure that the ones we have, we use in a way that they can be used for the longer term, and that means using them with moderation or in different combinations in a way that the bacteria doesn't become resistant too quickly to those antibiotics.
What would you say are the key findings from this study, and were there any that were surprising to you?
Yeah. So on the gonorrhea dataset that we looked at, the sort of key finding was that we were able to estimate exactly what is the fitness benefit and the fitness cost of resistance, and that allows us to estimate we could treat something like 10% of gonorrhea infection with ciprofloxacin without causing a rise in the rate of resistance of gonorrhea against ciprofloxacin. So even if it doesn't allow us to, we're not at all saying we should switch completely to this antibiotic. If we used it for even a small proportion, say 10%, it means there's no danger of that antibiotic, of the resistance to that antibiotic becoming increasing, but also it means it preserves the other antibiotics that are more commonly used at the moment, such as azithromycin or ceftriaxone, that are more commonly used and becoming at threat because they're being used so much, becoming at threat of not being effective anymore. Perhaps the most surprising finding was that we actually applied the method to two different lineages of gonorrhea that were resistant to ciprofloxacin, and that they are two sort of really completely separate lineages, and so we knew they were both resistant, but we would have expected perhaps that they had become resistant in different way, and the parameters might be different. But what we found actually is that the fitness cost and the fitness benefit of both lineages were exactly the same, or within the same intervals that we estimate. And that was a bit of a surprise because I would not necessarily have expected that to be the case.
Yeah, it's always interesting to hear when you get the unanticipated finding arising from when conducting such scientific analyses. And so given that, are there particular implications of, say, such a finding?
So the implication as far as gonorrhea is concerned is that we can treat, our suggestion is to treat up to 10% of infections of gonorrhea with ciprofloxacin, and we would suggest doing that at random, so that there are no sort of clusters of resistance arising. But more broadly, really, what we hope is that this algorithm will be used for other systems of bacteria and other antibiotics to really map out what are the fitness costs and benefits of resistance for a broad range of antibiotics and a broad range of pathogens. So tuberculosis, for example, is another bacteria that is increasingly becoming resistant to pretty much every form of antibiotics that we know. In fact, some strains of tuberculosis have even been described as totally resistant to any antibiotics that we know. So luckily, they are very, very rare at the moment. But so tuberculosis is certainly one system where we would want to apply those methods, and it's one of the sort of critical bacterial pathogen as far as resistance is concerned. Another good example that we previously worked on was MRSA, so that's a methicillin-resistant form of Staph aureus that caused a lot of trouble back in the 1990s, especially. It's pretty much sort of gone away to some extent now, but the MRSA crisis happened because there was so much beta-lactams being used at the time, and so that sort of created this monster of a pathogen, and it was only the only reason why it sort of went away is because people have been more careful in the way they use beta-lactams now. And so that's another good candidate for which we can really evaluate what are the dynamics of resistance, what are the fitness costs and benefits, and conclude from that what exactly is the best way to deploy the antibiotics that we have at our disposal.
So this would mean you can run in real time this algorithm, and it can guide you the proportions of, for example, antibiotics you can use.
In near real time, I would say. I mean, it's not an algorithm that you would want to run completely in real time, because it's not like we're not going to learn something really new from day to day. But you would want perhaps to periodically, maybe every month or so, update your set of genomes that you have available for analysis, and track what are the sort of dynamics of resistance. Are there any new lineages arising at the moment that seem to be of concern, exactly as was done for COVID with a variants of concern, and what are their properties of resistance, and what should we do about them in terms of antibiotic stewardship.
Looking ahead, what in your view are going to be the key challenges for pathogen genomics, as efforts are continuing to be made to mitigate the risk of antimicrobial resistance? And how may the Health Protection Research Unit in Genomics and Enabling Data contribute to this research?
So the main challenge that we have at the moment is in terms of making the most of the data. So the genomic itself, the process of sequencing the genomes, has made fantastic progress between 2000 and now, with some really, really efficient methods of sequencing, really that can sort of very quickly and also very cheaply we can sequence hundreds or thousands of genomes of bacteria, and so get a really good genomic surveillance program going on. The real bottleneck is, once you've got all this genomic data, what do you do with it? And so the mission of our Health Protection Research Unit in Genomics and Enabling Data is to develop that pipeline, so in partnership with UKHSA, especially one of the sort of main partners doing surveillance in the UK on resistance of bacterial pathogens. We want to develop the new methods of such as the one that we've talked about today, but also many other methods to tackle some of the challenges around what do we do with data, how can we develop algorithms that are powerful, that can scale to the large amounts of data that we have at our disposal, and that can inform the control policies of the future.
Thinking like during the COVID-19 pandemic response, the different types of data stream that would become available. It's like primary healthcare, secondary healthcare, genomics, virology, from various different disciplines, and so it requiring a broad team to reasonably to interpret all these data sources and basically translate it for people in other disciplines so they could potentially go and use it in their own modelling work, etc. So I think this very much links into the fact a lot of the research being conducted is interdisciplinary in nature, and that's going to be a very, I think, important area in upcoming years.
That's very true, and I should add that genomics is not at all a replacement for traditional epidemiology. I mean, I think at some point, some people got really excited about genomics and thought this might sort of answer all the questions, and we wouldn't need to sort of do any sort of standard epidemiology anymore. And that's not true, because even with whole genomes of bacteria from cases, we're very often unable to say like something like who infected whom, trying to reconstruct a transmission tree or anything like that, because the rates of evolution of the genomes are not always sufficient to do that. So really where genomics really shines is as a complementary approach with the traditional epidemiological methods, which means we have to work with partners who are epidemiologists, public health scientists, etc., statisticians, computer scientists, etc., in order to sort of make the most of all the different strands of data that we have to really inform control measures.
I have one question regarding data. Is it now more available for multiple pathogens to do routine basis genomics?
It is, as there's more and more genomic data available. In a country like here in the UK, we have a lot of good genomic surveillance program in place. So where it becomes a bit more challenging, and it's very relevant to resistance, in fact, is that, of course, what often happens is that resistant pathogens arise maybe in Asia or in Africa, and of course those countries don't have such good surveillance programs. And so what we see here in the UK is only the imports of those events, and to really understand how those new lineages arose, we would need good surveillance programs in other countries as well, or throughout the world. Another sort of example of that is when pathogens actually come from animal species, so then suddenly there's a lot less data available if on bacteria causing disease in animals, and so that can create the same type of issue where we see the imports of the resistance form or the variant forms of a bacteria in humans here in the UK, but we know a lot less about exactly where did they arise. Was it in an animal host? Was it in a different country, etc.? So yes, there's a lot of data available here from human infections, but a lot less here in animals, and even a lot less from other countries.
That's great to hear. Thank you very much for all the explanation you've given us today.
It was a pleasure. Thank you very much.
Very much looking forward to seeing the future work that's conducted.
Oh yeah, there will be more, yeah.
Thank you all for listening, and we hope you'll join us again next time for our next episode of SBIDER Presents.